share my screen. Excellent. So as you know, I'm going to be talking about the behavioral and cognitive effects of low to moderate alcohol exposure using evidence from both human and animal studies. I need to declare that I have nothing to declare. So moving forward, I'm going to be using the term prenatal alcohol effects because I'm going to be talking about both human and animal studies. But across the human literature, the most common terminology we use for children who has changes or impairments due to fetal alcohol exposure is the umbrella term fetal alcohol spectrum disorder. So this is a relatively more recent term. Previously in the literature, so the first study actually that came out looking at or describing negative fetal alcohol effects came out in 1973 by Jones and Smith. It was just a very brief communication saying, hey, you know, we've noticed that when children are born to mothers who drink a lot during pregnancy, we're seeing that they have these range of impairments in social, physical, and behaviorally. And this was followed up by a slew of studies over the last 40 years looking at the effects of alcohol. So the first syndrome that came to be associated with these effects was fetal alcohol syndrome. So this is one you've probably heard the most often. And as more research is done, it was found out that children actually show a whole range of impairments or a whole range of difficulties following fetal alcohol exposure. So the term fetal alcohol spectrum disorder kind of encompasses both high impairments or high changes due to the fetal alcohol exposure, and also more maybe just behavioral effects of this. So these include criteria or terms such as alcohol-related birth defects, which refers to just physiological changes associated with prenatal alcohol, and then the term alcohol-related neurodevelopmental disorders, which was, I believe, a diagnostic criteria that was used to differentiate children who had fetal alcohol exposure, but didn't show the same physiological changes associated with fetal alcohol exposure. And you see this diagram here. So this is common in first-year psychology textbooks when they talk about, you know, fetal alcohol syndrome or fetal alcohol spectrum disorders, of the characteristics that are used to identify fetal alcohol exposure in infants and children. And currently, for the Canadian guidelines for diagnosing FAS in infants and children, they have to show 3 of these features. So these include the short palpable features. So this is the actual opening of the eye. These children show a short nose, a flat mid-face, a thin upper lip, and an indistinct philtrum. And the philtrum is the bumpy bit from your nose to your lips. So if a child shows 3 of these kind of 5 criteria, they're put into a high-risk category for fetal alcohol effects. So with animals, however, they don't get this diagnostic criteria. So a lot of the animal literature uses the term fetal alcohol effects or prenatal alcohol effects, or even just early alcohol exposure. Now, in the last 40 years, we've learned a lot about the effects of heavy prenatal alcohol exposure. So these are the effects that we see in children and adults who are born to mothers who drank heavily and consistently throughout their pregnancy. So what we know from these studies is that there are definite cognitive impairments in these children. So these range from deficits in full-scale IQ. A lot of these children who have heavy exposure are classified in the mental retardation range. We see problems in kind of all domains of learning, but particularly in reading and arithmetic behaviors. In terms of problem behaviors in the home and at school, hyperactivity and inattention are very high in this group. And it's believed over 50% of children with FASD have a comorbid diagnosis of ADHD. So a lot of behaviors are similar between these 2 groups. We also see increased levels of aggression and poor behavioral control in these individuals. Socially, we see a lot of difficulties in these children as well. So these range from difficulties perceiving and responding to social cues. We see a lot of differences or difficulties in these children when they're in more complex social behaviors or more complex social situations. So if they're in a group with their peers, when they're very younger, we see differences in play behavior, so problems with reciprocity. We see this as they get older, they have difficulty taking turns in conversations. They have difficulty interpreting and responding appropriately to social cues. They can be very friendly with strangers. And it's seen that on the emotional end, they actually see that there's an arrest in their emotional development. So they don't show the same emotional behaviors, emotional intelligence as their same age peers. So some of these behaviors are very similar to what we see in children who have ASD, who have autism spectrum disorder. And ASD is actually also very highly comorbid with children who have FASD. So as a result of some of these social deficits, these individuals with FASD are more likely to experience bullying and victimization, and it's found these behaviors become more pronounced with age, particularly as they move from childhood to adolescence, where social behaviors become much more complex. Now, what we need to keep in mind anytime you're looking at prenatal alcohol exposure is you need to keep in mind what was the timing, the frequency, and the dosage of this exposure. So timing refers to the gestational time point, or what was the where was the fetus or embryo in development when alcohol was being exposed. We also have to look at the frequency of drinking events and the amount of alcohol consumed during each drinking event as well. So all three of these tie together to affect the impairments or the behaviors we see in the child. For example, you might have a mother who enjoys her wine Wednesdays, was 2 glasses of wine every Wednesday. You might have a mother who has much heavier binge drinking on a weekend, and you also have women who are heavy binge drinking kind of throughout the week. So some of the challenges of studying fetal alcohol exposure or prenatal alcohol exposure in humans is that this is very dependent on maternal self-reports of drinking. So if we really want to understand how alcohol affects, let's say, social behaviors in children, and specifically how low-dose alcohol affects social behaviors, we need detailed information about how much alcohol was consumed and at what developmental time point. And if maternal self-reports are often collected retrospectively, it's hard to get the level of information that is necessary to draw really strong conclusions. Children with FASD, particularly those who have higher alcohol exposure, they have a high percentage who end up in adoptive or foster care. So in these situations, maternal histories are difficult, if not impossible, to access. And when we're looking at human behaviors, as well as, we need to keep in mind, of course, the frequency, duration, and developmental time points of these alcohol exposures. We also need to keep in mind the interaction with genes and early environment as well. So if you're taking a child who's 5 years old and looking at their social behaviors, and you want to relate it back to known prenatal alcohol effects, you also have to take into account what their early childhood environment was like as well, because we know that different environments can also influence the same behaviors we want to look at in relation to alcohol. So one solution to kind of teasing apart all these differences is to look at animal models. And animal models give us control over many of these challenges that we see in the human behavior. And so in particular today, I'm going to be talking about low to moderate alcohol exposure, because this is not covered very frequently in the literature. So a lot of what we know about the negative effects of alcohol come from very heavy studies. But we know from other reports that most women who drink during pregnancy don't drink a very high amount. Not everyone is an alcoholic who's gestating. So we need to have better information on what low dose to moderate alcohol looks like in the human population, because this actually might be more prevalent. So we need a better way of identifying it. So the best way to identify is to learn more about it. So for animal models, I'm going to first talk about a zebrafish model, because this was the work that I did, so the one I'm most familiar with. Now, I know that fish are very different from humans in a lot of ways and different from mammals, but they just do share a lot of similarities. So about 80% of our DNA is similar between fish and humans. They're social animals. They show socially complex behaviors, and they're also very easy to manage alcohol dosing. So in this very simple schematic here, we have little eggs, and inside each egg we have a teeny tiny zebrafish embryo. So when I picked my developmental window you want to target, the eggs are just placed directly into an alcohol bath containing the amount of alcohol we want to expose them to. And the benefit of fish is that the alcohol goes through the egg membrane, goes right into the developing embryo, and we can directly measure how much is reaching the embryo. And we have less variability than you have in mammals, where you have to take into account like how much alcohol is being absorbed by the mother's liver, how much is actually passing through the placenta. And we know that even within a litter, or even with twins, there's different amounts of alcohol release reaching each fetus in a litter, for example, for rats. So we don't have the behavior with fish. So we give them their dosing, and for my particular paradigm, I looked at adult behaviors. So we waited 3 to 4 months, controlled the environment for all of them, so we could test just the effects of alcohol on various social behaviors. And this, my window, is just an example of the benefits of using an animal model and what we can really target with it. So this was an example of looking at just swim speed in fish. So we had two strains of fish. We had an AV strain, which is the ones I used, and then wild type on the bottom. And what we did was that every 8 hours, the fish were exposed to alcohol. So different groups were exposed to alcohol. So this group was exposed for 2 hours at developmental stage 8 versus 16, 24, and so on. So what we see if we're looking across just this one strain, the black boxes represent the alcohol-exposed fish that has 1% alcohol exposure, and then the bottom is our control fish. What we see is across each time point that we're looking at, we see an increase in swimming speed in our 1% fish compared to the controls. And the largest difference between the two groups is happening at the 24-hour time point, which is actually what I use for my social behaviors. But if we're looking at the wild-type fish, which is a different strain, we don't see this clear change or clear difference in behavior between alcohol and a non-alcohol exposed fish. So this just shows that we can use a very small amount of alcohol at one very specific time point and still see significant differences in a behavior in an adult fish. These are persistent changes that we can measure from this one-time alcohol exposure. So this, my graduate work was all focused on social behavior. That's where we're going to start for today. So social behaviors in fish, so they are complex. And if you've ever been to a pet store and you've looked at like the tank of minnows, you'll notice that it's really hard to take one fish and track that one fish as it swims around with all the other fish. So the best way, one of the easiest ways to measure social behavior in a fish is to have your experimental fish responding to other fish in a kind of contained habitat. So one way to do this is we have our experimental fish in a tank, and then we have two computer screens. So what happens is the fish swims around for 10 minutes, and then animated fish are shown on the screen. And zebrafish are highly visual species. So what we see in our control animals is we see they're swimming along, and then as soon as the stimulus is shown, which is the black bar here, we see them decrease their distance to the screen. So they're actually swimming right alongside the screen, which you see with like your little fish moving here, for the duration of the social stimulus. And then once the stimulus is turned off, we see the fish swims away. If we look at our medium concentration group, we see the same approach to the social fish, but they're not approaching as much as our control fish. And we look at the 1%, we're still seeing this dip where they are responding to the social stimulus, but they're definitely not showing the same preference for the social stimulus as we see in our control group if you compare the two. Another way to look at social behaviors with live fish, so this is a very similar paradigm, except we have live fish on a tank beside the fish on one side and then an empty tank here. So what we're measuring is whether they spend time near the social fish, whether they spend time in the center of the tank, or if they're spending time next to the empty compartment. And what we see here with the black bar representing our controls and the white showing the highest exposure group is we see this dose-dependent decrease in how much time they're spending next to the conspecifics. So our control fish are spending a lot of time right here next to the live fish, whereas we're not seeing that in our highest alcohol exposed fish. If we look at how much time they're spending near the empty tank, so in this area here, we see significantly more time being spent in our higher alcohol concentrations compared to the control or low doses. And this graph here just shows the breakdown of where they're spending their time in the empty tank, the center, and the conspecifics. So whereas in this paradigm here with the animated fish, we're seeing a decrease in social preference, we're actually seeing more of a social avoidance, like actively social avoiding the conspecifics when we're using live fish in this study. So we have a very clear change in social behavior in adult fish from this very low dose of prenatal alcohol exposure. Now in rats, we can measure similar behavior. So this is a social investigation task where you have our alcohol exposed fish, and then you have a naive animal that's never met before, that's not exposed, and you measure how the rat responds to the other animal. So for this study, they looked at a 1-day time point. So they took a time in like second trimester gestation for the rats, and they looked at how they respond to a novel animal at early adolescence, late adolescence, and in early adulthood. So what they found for males, with the black bars representing the alcohol-exposed rats, is there's this clear decrease in social investigation compared to the control animals, whereas in females, we only see this decrease in later adolescence and adulthood. And if we look at play behavior, so play behavior is very important for many social animals, rats in particular, or rats as well. And we see a typical age-related decrease in play behaviors in rats, just as you see in people. So we see no difference in males for this particular behavior, but when looking at females, we're seeing an age-dependent, we see a decrease in play behavior, more extreme than that's expected by age for the females exposed to alcohol compared to controls. So what we're seeing from this particular paradigm is that social behaviors can be altered in animals from a one-time dose in rats, and that we are seeing specific sex effects, and that time points can matter. So just similar to humans, adolescence in rats is a time when social behaviors become more complex because they change from play behaviors that we see in juvenile animals to more complex issues with aggression and territoriality and mate preferences in rats. So this is another study done by Holman et al. So this was looking at more moderate alcohol exposure. So instead of a one-time dosing, for these animals, they had alcohol exposure throughout gestation. And it should be noted that gestation in a rat compares to humans as about like the first 2 trimesters. So for rats, we consider the nursing period after birth, the 2 weeks after birth, as kind of the human equivalent to the 3rd trimester. So for this particular dosing regimen, they looked at kind of a human equivalent of a 2 trimester alcohol exposure. So similar to the last study, they looked at social motivation in these animals and found no difference between the alcohol group and the non-exposed group. And then they performed a social recognition memory task, which is when you have your 2 animals, your alcohol-induced animal becomes more familiar with one rat, they take them away for a period of time, and they introduce a new rat to see how they respond to this new rat. So what they found is that in early adolescence, we see a marked difference over time with how the control animal, which is the white bar, responds to the familiar rats. We see there's more time being spent with the novel rat by the control animals, but not with the alcohol-induced. And then we see the same behavior when they see the novel rats. We're seeing this decrease in social behavior in the social recognition memory task for the alcohol-exposed rats. This is really only most significant in the early adolescent period. They also follow this up with a social discrimination task. So this is again more complex, where the rat gets to know this one rat, they're paired again with the same rat that they know, and then the novel rat is introduced at the same time, and they look to see if they can make a discrimination between the 2. And what they find is that in terms of play latency, that they're seeing that there is an impairment happening across the 2 time points and in the alcohol-induced rat. So this longer, more moderate alcohol exposure is impairing much more complex social recognition and social discrimination tasks in rats. Now, social behavior in humans for low to moderate alcohol is a bit harder to find in the literature. So part of this is that the same behaviors that exist, or to say the paradigms that exist for rats. So for example, we have multiple paradigms in rats for testing social behaviors. We have different paradigms for fish. For any animal species, you can contrive a situation to really test specific aspects of animal behavior. And these don't translate as well to humans, maybe because most parents don't want their child to be treated like a rat in that kind of artificial situation. It's also much harder to control other behaviors when you have these complex social interactions in children. So we don't measure it in the same way. Most of the information that we do know about social interactions in children, with low dose in particular, low dose to moderate prenatal alcohol exposure, comes from parent reports, such as using the child behavior checklist, which is very common. So this particular study that actually did measure or look at social behaviors in children was this very large meta-analysis done by Flack. So they included studies, children from over 34 studies. It was almost 12,000 children to look at the effects of mild to moderate prenatal alcohol exposure. And they particularly were looking at studies that control for socioeconomic status, parental age, and IQ, as well as gestational smoking. So these are all things that we know have an impact on social behavior and other problem behaviors in children. So what their main takeaway from this was they only found differences in social behavior in younger children. So this was children that were 5 and under, and that there was an increased demand for attention by the children. They showed poor interactive play skills. So this is coming back to similar to the reciprocity problems and social cue problems that we see in more heavier prenatal alcohol children. And they also reported increased behavior regulations. And the main takeaway the authors concluded from this very large meta-analysis was that this mild to moderate encompasses women who drank less than 1 drink a day. So that might be sufficient to produce these impaired behavioral effects that we see in these children. And there was another smaller study, a cohort study done by Olson in 1997, so a little bit older. But this is a cohort from the US where they follow these children actually follow the mothers from gestation, so mid-pregnancy onwards until the kids were in their teens. They found in 14-year-olds, they had them do a self-assessment. So this was a self-report on how the teenagers viewed their own learning and their own behavior, and they had the parents fill out the child behavior checklist. So what they found was for moderate prenatal alcohol exposure, which they quantified as less than half a drink per day during pregnancy, was associated with increased self-reports of antisocial behavior and substance use, while the parents reported increased delinquent behavior, particularly fighting with other children, as well as substance abuse. So what this study found was early exposure, so exposure to alcohol that was happening in the early pregnancy, so during the first trimester, was more likely to be associated with negative outcomes than if alcohol exposure happened later in gestation. So what we're seeing for the humans for social behaviors is we're not seeing the same strong effects of prenatal alcohol exposure that we've seen in the heavy prenatal alcohol studies. But also there's a lot less being done looking at social behaviors, particularly in this low to moderate alcohol exposure group. Now, the next kind of category we're going to talk about are behaviors associated with anxiety and aggression. So starting again, of course, with fish. So one study that I conducted was looking at predator responses. So looking at a live fear response in fish who had the same very low dose of alcohol. So what we can see in this graph is these are control animals. This is behavior in the tank and how they respond to the predator. So we're seeing this active increase in escape behaviors from the predator, and this is the same across all concentration groups. So we're not seeing a difference in how these fish are responding to a live predator situation. However, when they're put into a novel tank to look at anxiety-like behaviors in these fish, what we typically see in fish who have high anxiety is they will sink to the bottom of the tank. And then they'll stay there for a while, and then they'll start swimming. So what we see in these animals is our control animals are in the black bar. The latency for them to swim to the top zone is significantly higher in our 1% or highest concentration group compared to controls. And if we look at the amount of time the animals are spent at the bottom of the tank, we see significantly increased times that they're spent in the bottom in all the alcohol groups compared to the control groups. And similarly, in line with this too, we see the distance to the top is also significantly less in the alcohol exposed group. So what we see here is there's actually an increased anxiety behavior seen in these fish from this one-time dose. And in rats, a lot of studies have been done looking at anxiety in rats. So they're often used to study the effects of anxiogenics and anxiolytics. So there's a lot of behavioral paradigms that exist to look at anxiety-like behaviors. So this one study by Cullen used moderate levels of alcohol throughout gestation. And what they used was an elevated plus maze, the whole board and eversion test to look at anxiety in rats. So for those who aren't familiar so much with animal studies, the elevated plus has these two arms which are covered, and then these two open arms. So an animal that has higher anxiety-like behaviors will spend more time in these covered arms. So in this example here, this rat is not showing very anxious behaviors. He's exploring right to the edge of this quite high maze. The whole board is these holes are in the board at the bottom of the maze, so an anxious rat will spend more time with kind of his head poking down. And in this task here, or this test, there's this covered tube in the center, so the animal who's more anxious will spend more time hiding inside the tube than a less anxious animal. So what they found was following this moderate dosing regimen is that animals showed higher anxiety in every single paradigm compared to the control animals who had no exposure. And this really supports what we're seeing in human literature. So this study, it was from Japan. It's very recent, and it's interesting in that it was actually a quasi-experimental study. So what they did was they have this large cohort that they follow throughout life. And from those, they picked women who had at least 2 children, one of which was exposed to alcohol prenatally, and the other one which had no prenatal alcohol exposure. And they only selected women who had either rarely drank during pregnancy or who had 1 to 4 drinking events per month, and to run these comparisons. So we have a very specific study set up here to look at specifically the effects of low alcohol exposure And to control, because they're using a sibling study, they can actually control for some confounding variables that we see in other studies, such as parental education, family income, domestic violence, and working status. So these are all variables that we know can influence the behaviors that we're interested in. So what they did was they took the child, let's say child A, who had prenatal alcohol exposure, and compared them to behaviors on the child behavior checklist with a sibling who was unexposed. And if they have multiple children, they took the one who was exposed and compared them solely with children who were unexposed. So what they found overall from this study, where over 300 sibling pairs had some prenatal alcohol exposure, was that this low exposure was associated with more anxiety problems and higher internalizing problem scores on the CBCL. They found increased aggression and attention, but only in the male siblings on this study. And if we're looking at anxiety and aggression, another study by O'Leary out of Australia, and this was another quite a large study, another cohort study found that moderate prenatal alcohol exposure during early pregnancy specifically was associated with increased anxiety in children, whereas moderate prenatal alcohol exposure during late pregnancy, but not early, was associated with increased aggression in children. And what was particularly interesting about this study, though, was they actually characterized alcohol consumption by the amount of alcohol consumed in grams, not just by 1 to 2 drinks. And what's interesting with the Australian medical guidelines for women who are pregnant advise that women have no more than 1 to 2 alcoholic drinks per day, and that they cap their limit of alcohol drinks at 7 per week, which seems very high considering that in Canada, the recommendations for a non-pregnant woman to drink during a week, I believe, is now like 4 drinks a week. And as the authors pointed out, for a woman who is drinking just 1 drink a day, as per these Australian health guidelines, they would already be placed in this moderate alcohol group. So we're seeing here that anxiety and aggression seem to be related to prenatal alcohol exposure, and there might be key developmental time points for when these behaviors manifest. And then a following study by Dosage is supporting these behaviors. So they had 500 parent-child dyads, and they found that, again, using the CBCL, that aggressive behaviors significantly increased just following low alcohol exposure compared to non-exposed control children. And then we also see increases in delinquent behavior compared to the non-exposed group, with obviously much larger variability and much larger averages in these behaviors in our heavier alcohol drinking groups. So finally, I'm going to talk about cognition, the effects of cognition following such early exposure. Now, in fish, to study learning and learning impairments, they did a plus maze task. So in this task, the zebrafish is placed in the middle, and then in only one arm, there's a red cue card placed at the end, and then there is a red food dropper. So there's food reward paired with this red marker, and the fish get trained on this for weeks. And after several weeks of training, the food reward is removed and only the red probe is left. So they look at how much time the fish spends in this target area, so right beside the probe where the food would be, to see if the fish learns this association task. What we see on the graph, this horizontal bar here represents chance responding. So we see in our control animals, they're significantly above chance spending time in this correct region. So we're seeing that, yes, they are learning the association between the probe and the food reward. But if you look at every single concentration group, they're not showing above chance performance levels for this task. So they're not learning the association between the food reward and the arm on this particular maze. Now, it should be noted too that this is, out of all the fish studies, this one was actually done at a different time point. So the earlier fish studies I discussed were all exposed at a 24-hour time point, and this was exposed at a 16-hour time point. So what was found is when the same study was done with fish who were exposed at 24 hours, when we see social impairments, they don't show learning impairment. So it's making the case where we have these very key developmental time windows for when, if alcohol is exposed during this particular time point, that's when we can see these negative effects or impairments. Now, this is a recent study done by Sanchez looking at low-dose alcohol in rats and also looking at learning discrimination tasks. So the first task is relatively simple. The rat is placed in the circle. It runs down an arm, and then it enters a chamber. And in the chamber, there are 2 toys. So here we have a black falcon tube and a red wooden square. And if the rat pushes the square, it gets a food reward. So what happens is this happens multiple times a day, and then we look at the trajectory of the correct responses. The dotted line here represents chance, and what we can see with our red being our alcohol-exposed rats, and then the blue being the control, is they both show that they're learning this discrimination task. So they can differentiate between the two objects, and then they learn which one gets rewarded or not, and they're performing at very high levels. The second task they ran was an object place paired associate task, which is a bit more complex. So similar to the first task, the animals start in the center, they run down an arm, and then they're in a chamber with two toys. One toy gets rewarded with food and the other one doesn't. However, if they run down the other arm, the opposite toy is being rewarded based on what place it is in the arm. So here, if they push over the Superman, they get food, but if they push the Superman here, they don't. So it's a much more complex task, and it's complex for all animals. As we can see here, it's day 6, and then both the alcohol-exposed and the control animals are still only responding at chance levels. But as we look over time, we're seeing this increased trajectory in correct performances happening with our control animals, and a much more abrupt and lower performance happening with our alcohol-exposed animals. So what this experiment is showing us is that animals are capable of learning, but there are impairments from this early alcohol exposure, but the impairments aren't global. So it's not like every aspect of learning is impaired, but this complex task is showing where we can see differences between them. So with these, I'm showing two here with these animal tasks are examples of tasks that we use, but the literature for animals is a lot less variable than what we see in humans. So this is pretty, these simple learning tasks across animals with prenatal alcohol exposure tend to be able to be performed. And then across different tasks, when we start seeing more complexity across different animal studies, that's when we start seeing differences in domains that are affected by the prenatal alcohol. Now for cognition in humans, so a lot of studies have been done with heavy alcohol exposure in cognition in particular, because a lot of these individuals end up in remedial programs at school. They need special help with daily tasks, daily living. So cognition has been a very big focus of that. So this is a study by Streifkus that came out several years ago. However, it was part of their cohort longitudinal study, and this particular time point they chose was 7-year-olds. So this is when children had already been in school for 1 to 2 years, so they're around grades 1 or 2. And they looked at moderate drinking, and they defined moderate drinking as 2 drinks a day, or actually they called it social drinking. But this was their moderate group, which might not be so moderate, depending on how you're classifying it, but it is in line with the moderate drinking as the Australian study we saw. So what they found was just 2 drinks a day was associated with a 7-point increase or decrease in total IQ scores in the children who had the exposure compared to the controls. And they found that in particular, the digit span and the arithmetic subscores were the most vulnerable. So these were 2 areas that showed the most significant decreases in relation to the alcohol exposure. For parent reports, the parents reported that these children had more difficulties reading compared to controls, and there was much more placements into remedial courses and special education courses than children who didn't have this. And what the authors note is that there are persistent attentional problems, specifically in this cohort. So what persistent attentional problems refer to is when these children were tested prior at 4 years of age, and when they were tested as infants, they showed attentional problems the whole way through. So this isn't a behavior that these children seem to be growing out of. And then this is a very recent study by Gao, and it's a much smaller study, but it's looking at cognitions in teens on specific tasks. So they classified prenatal alcohol exposure as either occasional to look for moderate effects or constant prenatal exposure to look at more heavy effects. And they found that regardless of whether they had moderate to high prenatal alcohol exposure, both groups displayed significantly lower total IQ scores compared to the typically developing teens. And they gave them a series of cognitive tasks to do. And the only task that showed the most significant decreases or most significant impairments in the occasional prenatal alcohol exposed groups were on the Cambridge gambling task, where these children showed higher impulsivity, and then they had longer response latencies on a shift set task. So what we're seeing across these cognition studies is mimicking kind of what we're seeing at the higher prenatal alcohol level, is that they're showing decreases in total IQ scores, and that we're seeing decreases in specific domains, we're not seeing global impairments, which is what you'd expect also too with having much lower exposure. Now, I know I've covered a lot in a short amount of time. I've showed studies from 3 different species. I've showed different behavioral paradigms, but the main takeaway from all of this, from our animal studies, is that we've seen that there are significant detrimental effects from a single exposure to alcohol. So we saw this across all the studies with the fish, and we saw this in the social studies too with some of the rats. For socially, we saw that we have impaired social behaviors, particularly as social situations become more complex. There is increased anxiety in both animal models that we looked at, and we saw impaired learning performances. And then in the rats in particular, we can tease apart differences in different domains where we see learning impairments compared to more simple tasks where we don't see that impairment as much. For human studies, for low to moderate, and I'd probably lean into the more moderate aspect for the studies that we talked about today, but social behavior, which is less well studied in this exposure group. But we see poor interactive play, more behavioral demands in the younger children as reported by their parents. Teens themselves report more antisocial behaviors themselves, and we see more delinquent behaviors as reported by their parents. We see increased anxiety, even with low exposures from the Ishikawa study. And then in aggression, we also see multiple studies that looked at increased aggression in both just males and both sexes. And it may be the result of certain time points, so later exposure to alcohol during pregnancy might result in increased aggression in these children. In terms of cognition, we're seeing that there are dose-dependent decreases in full-scale IQ, and this seems to carry through to even higher doses. And there is domain-specific impairments, just based on some of the studies we see in teens and the studies we saw in the longitudinal cohorts as well. There are, of course, limitations, some of which I mentioned at the beginning with how we study humans. But one of the big ones across both human and animal studies is that currently there is no consensus for what constitutes low to moderate or heavy alcohol consumption. So even studies which are measuring like binge drinking versus non-binge drinking. Binge drinking in some studies is 4 drinks, whereas in another study, binge drinking is 8 or more drinks on one occasion. So it's really hard to do comparisons across studies or even draw conclusions about the effects of different amounts of alcohol because we don't have this consensus between them. So there's a scarcity of studies for both humans and animals looking at lower prenatal alcohol effects. And that makes sense from a realistic standpoint. So it's easier to draw participants from, for example, an FASD clinic where they've already been diagnosed. And if they've been diagnosed, they're much more likely to have more significant impairments than those who have more subtle effects. And for animal studies, it's a lot easier to study an effect that is really big and really obvious than to look at maybe lower doses and try to find more subtle but significant effects. For human studies in particular, there are many confounding factors that we just can't control for because you can't do this. You can't set this up to be like a true experimental design. And although we can learn a lot from animal studies, we can't always make a direct comparison to what we see in an animal study compared to humans. And part of this is just differences in gestation. Like it's not a one-to-one relationship between gestation in a rat and gestation in a human. These are all things we have to take into consideration. But overall, the main conclusions are that mild to moderate exposure can and does lead to impairments in some domains, even if these changes are mild. We do see significant changes. There's evidence that even 1 drink per day can have negative effects on the child, and these effects are long-lasting. We see them in teen years, and the animal models, we even see them carrying through to adulthood. So the main takeaway from everything is that so far there's no evidence to support that there's like a safe amount of alcohol consumption during pregnancy. And that is the last thought that I will leave with you with. So thank you.
Diane, that was fantastic. Thank you. That was a nice summary of the work you've been doing. And it's really fascinating. And I asked you once about, first of all, who decided to use a zebrafish, but also what the advantages were. And I think that you explained it really, really nicely today as well. So we have lots of time for questions, which is great. Claire, I don't like to put people on the spot, but if you don't mind, I will put you on the spot. Do you have any thoughts or comments? Here, let me unmute. Okay, yep. Should be okay now.
Okay, I hope I'm unmuted. I'm on a very complicated setup here. So thank you, Diane. That was a great overview. Yeah, I think it's so helpful to have the animal studies to look at some of the neurobehavioral outcomes in the absence of any of the psychosocial factors. And I think anxiety is something that parents really see in their kids with prenatal alcohol exposure, even in optimal environments. And so I think that work is very interesting. I guess I'm wondering if there are studies looking at what we would consider really low dose alcohol. So less than a drink a day. So someone who, you know, might have a couple of glasses of wine or something in a week, a number of weeks through pregnancy. Is there anything that has looked at, or do you have animal models that look at that really true low dose alcohol, but persistent through the pregnancy?
I don't know any that model that specifically for animals. I know what you mean. So you're looking at maybe we have a woman who drinks, you know, she has one drink 2 or 3 nights a week. So there's no animal model that mimics that. Part of the issue is that gestation in animals is so much much speedier, right? Like we don't have 9 months in an animal to space apart that dosing. I'm trying to think off the top of my head what would be the best, what comes closest to looking at that. And I'm drawing a blank currently, but if something pops up during the question period, I'll shout it out.
You know, I guess the population from a clinical perspective.
Claire, is it possible for you to talk closer to your microphone?
Oh, yes. How's that? Is that a little better?
Slightly.
No. Let me just unplug from my screen. How's that? Is that better?
Sure, minimally.
I have to turn that off. Here we go. How's that?
Oh, fantastic.
Okay, hang on. Maybe I should let someone else talk for a second.
Okay, so while Claire's doing that, there was a question actually from Jennifer McLean about, Diane, do you have any hypotheses about why earlier alcohol exposure may lead to problems with anxiety, but later exposure might lead to problems with aggressive behavior?
Specifically, no. So there are, the problem is looking at the mechanisms of why these behavioral changes happen. So there's a lot of studies, particularly in animals, looking at so what's happening in the brain at specific time points that can relate to these behaviors. So example, for studies that look at social behavior, we know that there's changes in like the oxytocin system and the dopaminergic system, which really tie into social reward and social preferences, particularly in animals. And there's specific time points during development when we can, you know, alcohol has a greater effect on how these systems are developing. Specifically for anxiety versus aggression, I'm not sure. Studies that look at oxytocin haven't found changes in vasopressin, which has also been related to aggression in some models. So I'm not familiar with why this could be the case. And also there's a lot less mechanistic studies being done in humans, just because we can't pick around in there as much as there are with animals. So there's less, the aggression, what I talked about for the aggression in humans isn't as well supported in the animal literature. So they show more social avoidance. So even with social avoidance, they're not showing increased aggression because they kind of don't care about the other animal. So it might be something that's particular to those time points in human fetal development.
Okay, thanks. Claire, did you want to say now what you're saying?
Sure. So I just put it in the chat. So the clinical scenario is one where
a woman has alcohol as part of their lifestyle. They're not expecting to get pregnant. The pregnancy is diagnosed late, let's say 4 months gestation, and then they don't drink after that. So is there an animal model or findings for that scenario?
So there's animal models that look at, yes, just particular, that look at drinking kind of just first trimester. So even the model that I used was like a one-time binge, it'd be equivalent to, similar to like a one-time binge drinking event in a woman, kind of late first trimester. So that kind of time period you're talking about where, you know, she doesn't know she's pregnant yet, she goes to a party, and we see these resulting effects. There are human studies that found, I think it was by, one of the studies was by May, the researcher, and that even women who are heavy drinkers, if they stop drinking mid-pregnancy or decrease their drinking, they decrease their chances of having a child diagnosed with FASD by about 5 times compared to those who continue on with the drinking trajectory. So we know from the human research, even if you stop drinking later in pregnancy, it still has better outcomes for the child than if you continued on. And so with rat studies, though, most of them look at, there are some that look at first trimester or early gestation versus complete gestation in terms of drinking patterns in these rats. And we do see like dose-dependent impairments in these animals. But off the top of my head, I don't know which behaviors specifically they were targeting.
Thanks. Diane, are there any, so what you're talking about, how, you know, we know that so some people are going to have problems with anxiety or aggression. And what I've always wondered about, and you mentioned that we can't really control things, is genetics and stuff like that, and what role that might play in, and what happens to the fetus is exposed to alcohol. Do we know anything about the later life of people who are exposed to alcohol in utero? So do they have a higher rate of alcohol problems when they get older?
Yes, there are. So children who are exposed to prenatal alcohol are more likely than the general population to develop substance abuse problems. And this is shown in the rat studies as well. So the animal literature supports this. And it's, I believe, it's thought that prenatal alcohol exposure primes the brain to be more susceptible to the rewarding effects of alcohol. So when someone who has prenatal alcohol exposure has alcohol when they're a teen or when they're an adult, the rewarding effects or the pleasurable effects of alcohol are much stronger for them than they are for someone from the general population. So they're much more likely to develop a problem or addictive behaviors to the drug. And this is probably tied to changes in the dopaminergic system.
But how much of that is genetics? And maybe I'm asking you to speculate, let's say, but I mean, what role do you think genetics plays in that?
Oh, I don't know. So we also know that children who are born into families of alcoholics also are more likely to become alcoholics.
Yep.
So I don't know how to tease apart studies. I don't know of literature that's teasing apart the effects that are purely based on alcohol, but we do know from animal studies where they're not in a, there's no alcoholic mother and father raising these rats, and they're fostered by a different, like, sober family. We still see them that they have these effects based on alcohol. So in terms of, like, if you're asking there's specific genes that make them more susceptible, like a gene alcohol interaction, I'm not sure.
No, I'm not asking for your specific genes, but I mean, that's interesting with the rats, though, that if the offspring of the rats are more likely, so the offspring of the rats are more likely to develop alcohol problems?
Yeah. Well, the way they respond to alcohol, they become addicted to alcohol much quicker than control rats who didn't have prenatal alcohol exposure. And they show more of an affinity to liking alcohol because most animals don't like alcohol. You have to make animals be addicted to alcohol or drugs.
Well, that's because alcohol by itself probably doesn't taste very good for most people, but that's a different issue altogether. At least at first. Any thoughts or comments? What I also wonder about, Diane, is so what advice would you give to females of childbearing age about drinking alcohol?
So I'm not a lot of fun at parties. So I'm constantly telling people, you know, I get asked when people find out what I did my, you know, my Ph.D. dissertation on, like, oh, well, how much can I drink? And the answer was like, you should drink nothing. You should be doing nothing in terms of like things with alcohol. But the biggest problem is anecdotes, like anecdotal evidence, like, oh, well, my mom said she did and it was okay, or my friend said she did and her children were fine. But if you look at it, so the people who say like, oh, I drank and did drugs during pregnancy and my kid was fine, what they mean was your baby was fine. So your baby didn't show any obvious signs of health problems. But when you have a 10-year-old then who has, maybe they have behavioral issues, they have ADHD, and they have a lot of learning problems in the classroom, they're not making the relationship between what they did 10 years ago when they were pregnant and the behavioral and classroom issues their child is having now. So if you look, you have to look at it in the long-term effect is that, yes, you know, we do know that even a little bit is detrimental. But the problem is, you know, not every pregnancy is planned, and it's really hard to tell people in their teens and 20s that maybe they shouldn't be drinking on the off chance that they get pregnant.
Well, I guess that was sort of the group I was thinking mostly about, is how do we address that? Yeah,
so I was part of a group. I went to a conference in the Halton region where they were doing a specific ad campaign telling people women in their 20s that they should not drink because they might get pregnant. And I think they're trying to make the association with that. If you're having sex, you shouldn't be drinking alcohol, but these things often go hand in hand. So it's difficult. I know that Streiskup, so she's one of the pioneers of the FASD research, and she has FASD centers in Seattle. She did have outreach centers to try to prevent or decrease drinking during pregnancy. But I don't know I haven't heard of any plan that has worked to reduce drinking in people who aren't expecting to be pregnant.
Yeah, that's a great question.
Like it's kind of a hard, it's the category of people who are more likely to become pregnant unexpectedly and drink are people who are, you know, they're the age group to be heavier party drinkers. So it's challenging.
So this is Claire Mitchell again. There is an innovative project, I think it's in Alaska and in a number of other places where bars have pregnancy tests in the washroom.
Oh, wow.
So if you go out, you can just go in, do a pregnancy test. And if it's negative, then, you know, okay to drink that night. If it's positive, obviously not. And I think, you know, there are a group of partiers, but I certainly have talked to women who, you know, they graduated from university, they had a couple of parties to celebrate that, and then found out later they were pregnant. So I do think it's across all walks of life. And yeah, there's clearly more education is needed toward women of childbearing age to be kind of thinking about that, because I think, you know, eliminating alcohol consumption in that age group as a whole is probably not going to happen. But some of these harm reduction things like regular pregnancy tests are really innovative. So the other comment I had was I did read a rat study at one point, and I don't recall the author, but the rats who had prenatal alcohol exposure preferred to spend time with intoxicated rats versus non-intoxicated rats. And so I think there's a lot of studies looking at the behaviors and sort of poor judgment, I guess, if you can say that, about rats, which have similarities in human populations as well.
Yeah, so we know that, oh, sorry, keep going.
No, go ahead.
I was saying, we know that alcohol leads to changes in social behavior. So it's obvious in humans, right? If you see someone who's intoxicated, they don't make maybe the best social choices. So we see that across different species, that it really impairs their social judgment. In terms of rats preferring to spend time with intoxicated rats, what they found is when they have rats that are exposed to alcohol, they were thinking that, okay, you have a rat that maybe more have a predilection to having weird or different impaired social behaviors. So if you put them with other rats who also have impaired social behaviors, maybe you're just going to exacerbate the problem. So what they did is they cross-fostered. So they took an alcohol-exposed rat and they raised it with a litter of pups that were not exposed, thinking, okay, it's raised in like typical rat behaviors, so maybe we'll see differences in how it responds. And what happened was all the control rats started behaving like the alcohol-impaired rat. So we see this kind of social transmission of impaired behavior in the opposite direction of what they predicted. And we know for rats are neophobic, so they're afraid of new things, particularly new food, which isn't surprising given that there's so many poisons developed for rats. So one way they know that a food is safe is if they smell it on the breath of another rat. So if you give a rat a new food, they won't eat it. If they smell that food on the breath of another rat, then they'll eat it right away. So it could be that this mechanism in their brain that makes them more receptive to alcohol makes them more likely to be receptive to the smell of alcohol on the breath of an intoxicated rat. So that's interesting. I'm going to look up that study.
Jennifer McLean, a question. Diane, are there any protective factors found in animal models or studies that lessen the effect of alcohol exposure? And I must say that Ben Goldberg, who's not with us here today, couldn't join us, but he had a similar question because I think he said he'd read something about nutritional supplements or something like that that may have a protective effect. Is that correct?
So there are treatments that they're looking at that can, so the one in particular is choline. So choline during development is associated with the maturation and development of cells in the hippocampus. Animals that have an abundance of choline have better connections between hippocampal and the frontal cortex connections, and they show better performance on memory tasks. So what happens in what they've shown is in animal models of prenatal alcohol exposure, when you give them choline post-gestation, so postnatal choline supplementation, these animals actually recover some of these behaviors on these learning impairments. And they've actually done, I might have it here. They did choline, this was a double-blind placebo-controlled clinical trial for humans with choline. So they took 69 heavy drinkers, and in the middle of pregnancy, they gave them either oral choline or placebo, and they used idling conditioning, which is a habituation task. And what they found is that there's much better responding for the children who had the choline supplementation in pregnancy compared to those who didn't. And they even did choline studies in children who I believe were 4 to 5 years old. So some received choline for 9 months, some received placebo, and then they found a difference in age and exposure type. Whereas I think I can't see my screen. What do we have here? Those who have choline when they were young were performing at better levels than those who had choline, who had the placebo. But there was less of a effect if they received the choline when they were slightly older. So there are some things that it's not preventative, like it's not an endogenous preventative measure, but it's something that can be given to pregnant mothers or to children afterwards that can, in particular, it helps with learning impairments. And then in some, I think I read one study in a rat where it showed it also helped recover some social impairments.
But what if do you know of any studies that have been done using just regular rats who weren't exposed to alcohol, choline in the same time frames? So is this an effect that's specific to rats that were exposed to alcohol, or is it an effect generally? Or sorry, for children, I mean, sorry.
When they've given it to rats that were just control rats that didn't have alcohol exposure, they showed like increased memory performance.
So what I meant, sorry, was if they give choline to just newborns, and I guess ethically that might be difficult to get approval to do a study of, but is this effect that you see here, is that specific to kids with fetal alcohol spectrum disorder, or is it a general effect?
Would they all benefit from having choline? I don't know. So I haven't read much about choline treatment. So what was the other one? So this was 2018 and this was 2015. So it's relatively recent. And I haven't heard a lot about it. So I don't think it's a widespread treatment currently being used. And of course, I was thinking too, in terms of just like the ethics behind this, for this to work, particularly if the women are still pregnant, they're still drinking during pregnancy. So I'm obviously not a clinician, but you think you would want to first try to get the woman to decrease drinking during pregnancy, as well as administer choline, maybe to recover what has already been impaired.
Yeah, and what I would wonder, so my understanding from family doctors is that I think with some medicine, so with things like statins for cholesterol, that sometimes people see, oh, I'm taking a statin, therefore I can have a steak every day and it doesn't matter. And so I wonder if this was available, would that, I don't know if I'd want to say it would encourage people to drink more alcohol when they were pregnant, but would it decrease people's concerns about drinking alcohol in pregnancy?
Yeah, you don't want to encourage like complacency in something that has such serious consequences. So yeah, so there is, you know, even if we were to find like a safe amount of alcohol per se, like let's say, oh, 1 drink a week is okay. When you express that to the general public, do they actually know that 1 drink a week is okay? It's 1 drink of beer, not 1 drink of whiskey. Like, what's the difference? And like, this is very anecdotal. When my sister-in-law was pregnant, her doctor told her she could have 1 drink every other day in terms of alcohol, and her baby should be fine. And he told her 1 drink of wine. She reasoned that, oh, she doesn't drink wine, but she likes drinking rye or rum, so she can drink rum, you know, every other day. And she doesn't want to drink it during the week, so she'll just drink it 3 drinks on the weekend instead. So logically, like, I can see how she got there, but in terms of the actual science, it's just a bad outcome, or you're leading towards a bad outcome. So for what the science tells us and what we can, I think, safely tell the public about the effects, we have to, I think, be very careful.
Yeah, because you'd almost want to be like a certain drug companies and bury that kind of finding and just leave it as it's better to have no alcohol.
Yeah. So even in like, that's a takeaway from so many studies where they, you know, they say their goal was to find a safe amount of alcohol exposure, and the takeaway is always, you know, it's still looking like complete abstaining from alcohol is your best bet.
Okay, other questions or comments? This was a really fascinating talk. Really thrilled by it, Diane. Thank you.
Oh, thank you for having me.
So just a reminder, everybody, if you would like credits from either the Royal College of Physicians and Surgeons or the Ontario Psychological Association, then we need your correct name and email address. Sarah O'Flanagan will send people a questionnaire, an evaluation form tomorrow, and then also make sure that people get their credits. Diane, I'd like to thank you once again for joining us. This was a fantastic talk, and I really found it wonderful. So thank you again.
Okay, and thank you for everyone for coming up with this.